If you close this video right now, you will permanently lose the 5 specific foods that determine whether your brain enters its repair cycle tonight or continues accumulating the toxic protein deposits that neurologists now understand to be the primary biological mechanism behind Alzheimer's disease, being foods that feed a maintenance system operating in your brain every single night that Western medicine largely ignored until a landmark discovery published in Science in 2013 revealed that the brain has its own dedicated waste clearance infrastructure, an infrastructure that becomes measurably less efficient after 60 and that requires specific nutritional inputs to function at the capacity needed to protect your memory and processing speed. Inputs that the standard Western dinner for adults over 60 consistently fails to provide, while the same adults spend billions annually on memory supplements that cannot reach the biological target these 5 whole foods address directly. Japanese neuroscience integrated this discovery into dietary guidance by 2017. Your neurologist has almost certainly never discussed it. Most people over 60 who experience memory lapses, word retrieval difficulty, or mental fog have been told one of 2 things. Either this is normal aging and nothing can be done, or they should consider cognitive screening to rule out early dementia. After 26 years of clinical practice spanning Western neurology research and traditional East Asian integrative medicine, I can tell you that both responses miss the most important biological variable in the conversation. The cognitive changes that most adults over 60 experience are not primarily the result of irreversible neuronal death. They are primarily the result of a maintenance deficit, a nightly repair cycle that has the biological capacity to consolidate memories, clear toxic proteins, and rebuild synaptic connections, but that is operating without the specific nutritional tools it needs to do so. The difference between a brain that wakes up sharper and a brain that wakes up foggier is determined in large part by what was on the dinner plate the night before. My name is Dr. Jae Yoon. I am a physician trained in both Western clinical medicine and traditional East Asian integrative medicine, and I have spent 26 years studying why older adults in Japan, South Korea, and Taiwan maintain cognitive clarity, memory function, and neural processing speed at rates that consistently outperform Western counterparts of the same age. The specific variable that East Asian neuroscience and nutrition research identified, and that Western clinical practice has yet to translate into practical dietary guidance, is the role of evening nutrition in supporting the glymphatic system, the brain's own waste clearance and repair infrastructure that operates exclusively during deep sleep. What I am going to share with you tonight, before you eat your last meal of the day, is not theoretical. It is the direct application of peer-reviewed glymphatic biology to a practical dinner strategy that your brain is waiting for every single night. I want to stop for just a moment because I need to ask you something directly. I do not have a pharmaceutical company or supplement brand funding this channel. What I have is this research, this camera, and you. Right now, before we go any further, do 3 things that cost you absolutely nothing. First, press like right now. Without that, like, YouTube buries this video, and the person who needs it most never finds it. With your like, this reaches 100,000 people, and somewhere in that number is an adult over 60 who woke up this morning unable to remember the word they were looking for and has been quietly frightened about what that means. Your like is the reason they find this today or never do. Second, subscribe and turn on the notification bell. Every week I release research your neurologist was never taught to translate into dinner recommendations, and your subscription is what keeps this channel reaching people who still have time to act on it. Third, leave me a comment right now telling me what country you are watching from and whether you have noticed your memory or mental clarity changing in ways that concern you. I read every single one. Now let me explain what is happening in your brain while you sleep. In 2013, a research team at the University of Rochester published a discovery in the journal Science that fundamentally changed what neuroscience understands about sleep. They identified a previously unknown waste clearance system in the brain they named the glymphatic system, which operates exclusively during deep sleep by allowing the spaces between brain cells to expand by up to 60%, creating channels through which cerebrospinal fluid flows rapidly, flushing the metabolic waste products generated by waking neural activity out of the brain. The most critical waste product cleared by this nightly flushing is beta amyloid protein, the same protein that accumulates in the brains of Alzheimer's patients to form the plaques that disrupt neural communication and that most pharmaceutical interventions have failed to address from the outside. In adults under 50 with healthy sleep architecture, the glymphatic system clears beta amyloid efficiently enough to prevent meaningful accumulation. In adults over 60, efficiency declines measurably, and the rate of clearance begins to fall behind the rate of production. This is the biological mechanism behind the cognitive changes that most Western physicians describe as inevitable aging. It is not inevitable. It is a maintenance deficit with specific, addressable nutritional causes. The glymphatic system over 60 does not stop working. It starts working with insufficient materials, and the evening meal is the last opportunity to deliver those materials before the overnight repair shift begins. Understanding this distinction between a broken system and an underfed one is what changes the conversation from passive acceptance of cognitive decline to an active decision made at the dinner table every single night. The glymphatic system's clearance efficiency depends on the health and flexibility of the astrocyte cells that control the expansion and contraction of the interstitial channels through which cerebrospinal fluid flows. These astrocyte cells require specific fatty acids for membrane integrity, specific neurotransmitter precursors to regulate the sleep depth at which glymphatic activity peaks, and specific anti-inflammatory compounds to prevent the neuroinflammation that rigidifies the channel walls and slows fluid flow. When these specific inputs are absent from the evening meal, the repair shift starts understaffed. The beta amyloid clearance is incomplete. The synaptic consolidation of the day's learning is partial. The mitochondrial renewal in neurons that determines the next day's processing speed does not complete. You wake up tired, you wake up slow, and over years the cumulative deficit of incomplete nightly maintenance becomes the cognitive decline that most physicians attribute to inevitable aging rather than to a correctable nutritional insufficiency. The evening meal is the last nutritional delivery window before the glymphatic system's nightly operating cycle begins, making dinner the single most consequential meal of the day for brain health after 60, a fact that is absent from standard Western dietary guidance, but embedded in East Asian clinical nutrition practice for nearly a decade since the glymphatic discovery was published and replicated across 7 independent research groups. Research published in Nature Neuroscience confirmed that disrupting glymphatic function accelerates beta amyloid accumulation at rates measurable within weeks, while optimizing the nutritional inputs available during the repair window produces the inverse effect. This is not a marginal distinction. It is the mechanistic foundation for understanding why 2 people of the same age, sleeping the same hours, can have cognitive trajectories that diverge dramatically based primarily on what they ate for dinner. The first food is wild salmon, and its role in glymphatic function is direct and documented. Wild salmon is among the most concentrated whole food sources of DHA, the omega-3 fatty acid that constitutes approximately 40% of the fatty acids in the brain's gray matter, and that is the primary structural material from which astrocyte cell membranes are built. When DHA is sufficient in circulation during sleep, astrocytes can maintain the membrane flexibility required for the channel expansion that enables glymphatic flow. When DHA is deficient, which a University of Pittsburgh study found is the case in approximately 70% of adults over 60 consuming a standard Western diet, astrocyte membranes become rigid and glymphatic channels cannot achieve the 60% expansion that efficient beta amyloid clearance requires. Wild salmon additionally contains astaxanthin, one of the very few dietary antioxidants capable of crossing the blood-brain barrier, delivering direct protection to neural tissue during the waking day's metabolic activity. The second food is tart cherries, and their role is ensuring the brain actually reaches the deep sleep stages where glymphatic activity peaks. Tart cherries are one of the few whole foods containing measurable concentrations of naturally occurring melatonin, the hormone regulating sleep depth architecture. A study from Louisiana State University found that adults over 60 consuming tart cherry juice twice daily showed 84 minutes more sleep time and significantly improved sleep efficiency compared to placebo, with the improvement specifically concentrated in the slow-wave deep sleep stages where glymphatic clearance operates at maximum capacity. Beyond melatonin, tart cherries contain anthocyanins, shown in peer-reviewed research to cross the blood-brain barrier and directly reduce neuroinflammation, addressing the inflammatory rigidity in glymphatic channels that is one of the primary reasons the system slows after 60. Dried tart cherries consumed 2 to 3 hours before bed deliver both mechanisms simultaneously. The third food is whole eggs, specifically the yolk, and I want to address this directly because decades of Western dietary guidance warning adults over 60 away from egg yolks based on cholesterol concerns has created a widespread choline deficiency in exactly the population that most needs this compound for cognitive maintenance. The egg yolk is the most bioavailable dietary source of choline, the precursor from which the brain synthesizes acetylcholine, the primary neurotransmitter of memory formation and retrieval. During the deep sleep stages in which the glymphatic system operates, the hippocampus simultaneously performs memory consolidation, the transfer of experiences from short-term to long-term storage, a process that depends directly on adequate acetylcholine availability. Every pharmaceutical intervention for Alzheimer's currently approved by the FDA works by attempting to prevent acetylcholine breakdown. 2 whole eggs at dinner provide approximately 250 milligrams of choline, representing more than 40% of the recommended daily intake for adults over 60. The fourth food is walnuts, and their mechanism operates through a biological pathway not fully understood until research from the Salk Institute in 2016 identified the compound responsible. Walnuts contain ellagic acid and ellagitannins that the gut microbiome converts into urolithin A, a postbiotic compound that activates mitophagy, the cellular process by which aging, dysfunctional mitochondria are dismantled and replaced by newly synthesized ones. In the context of brain aging, neurons are among the most energetically demanding cells in the body, and the mitochondria that power them accumulate damage over time that reduces their ATP production capacity. Neurons whose mitochondria are not being regularly renewed through mitophagy become progressively less efficient at synaptic transmission, memory consolidation, and glymphatic channel regulation. Korean longevity research consistently identifies walnut consumption as characteristic of dietary patterns associated with maintained cognitive processing speed in adults over 70. The fifth food is spinach, and its role in nightly brain repair operates through 2 distinct pathways. The first is nitrate-mediated cerebrovascular dilation. Spinach contains more dietary nitrates per gram than almost any other commonly consumed vegetable, and these nitrates are converted by oral bacteria to nitric oxide, a vasodilatory compound that increases cerebral blood flow by relaxing the smooth muscle in cerebral blood vessel walls. During deep sleep, adequate cerebral blood flow is essential for both the delivery of nutritional substrates the repair process requires and the removal of waste products being cleared from interstitial spaces. Research from the University of Exeter found that dietary nitrate supplementation, equivalent to a standard serving of spinach, produced measurable increases in cerebral blood flow in regions most associated with cognitive function in adults over 60. The second pathway through which spinach supports nightly brain repair is folate-mediated epigenetic regulation. Spinach is among the richest dietary sources of natural folate, which serves as the methyl donor for DNA methylation reactions that activate and silence the specific genes governing neuronal repair, synaptic plasticity, and anti-inflammatory response during sleep. Karolinska Institute research tracking sleep quality and beta-amyloid clearance markers in adults over 65 found that dietary nitrate intake from vegetable sources correlated significantly with both sleep depth measures and lower cerebrospinal fluid beta-amyloid concentrations, providing direct evidence connecting the spinach pathway to the glymphatic clearance outcome, and confirming that a single evening vegetable serving can measurably influence the nightly brain maintenance cycle. This is the infrastructure component of the repair system, and spinach is the most accessible dietary source of both its fuel and its regulatory signal. The nightly timing of these 5 foods is the variable that determines whether their active compounds are in circulation at the biological moment when the glymphatic system needs them. The deep sleep stages in which glymphatic clearance, memory consolidation, and mitochondrial renewal peak occur between approximately 90 minutes and 4 hours after sleep onset. For an adult falling asleep between 10 and 11, the repair window peaks between 1 and 3 in the morning. A dinner consumed between 6 and 8 places DHA, choline, urolithin A precursors, anthocyanins, and dietary nitrates in peak circulation precisely during this repair window. A dinner consumed at 9 or 10 pushes digestion into competition with early sleep stages and delays peak absorption until after the primary glymphatic window has passed. Japanese clinical nutrition guidelines for cognitive aging specify the 2 to 4 hours before sleep onset as the critical nutritional delivery window, and this timing is the element most commonly absent when adults attempt this approach without complete results. The practical meal combining these 5 mechanisms is not complicated and requires no unusual ingredients or preparation. A dinner of wild salmon, 2 whole eggs, a large serving of fresh or wilted spinach, a small serving of walnuts, and dried tart cherries or a small glass of tart cherry juice taken 2 to 3 hours before bed covers all 5 biological pathways simultaneously and costs less per serving than the average memory supplement that addresses none of them. The synergy between these foods is measurable. The DHA from salmon enhances absorption of the fat-soluble astaxanthin. The nitrates from spinach improve the cerebral blood flow that delivers choline from eggs to the hippocampus during consolidation. The melatonin from tart cherries ensures the deep sleep architecture in which all other compounds deliver maximum benefit. And the urolithin A pathway from walnuts renews the mitochondrial infrastructure that powers every other repair process. The research foundation for these 5 foods in the context of glymphatic function and cognitive aging is consistent across institutions. A study from Rush University Medical Center in Chicago, following more than 900 adults over 60 for an average of 4.7 years, found that those consuming the dietary pattern most closely aligned with these specific food categories showed cognitive aging equivalent to being 7.5 years younger than their chronological age. Research from the University of Oxford following more than 500 adults with mild cognitive impairment found that dietary interventions targeting choline and omega-3 availability specifically produced measurable reductions in brain atrophy rates over 2 years compared to placebo. The pattern across these independent findings confirms that the specific nutritional inputs for glymphatic support, not general healthy eating, are what determine the rate of cognitive maintenance after 60. I want to address adults over 60 who have been told that dietary intervention at this stage is unlikely to make a meaningful cognitive difference. They are being given an answer based on the state of neuroscience before 2013, before the glymphatic system was identified, and before the relationship between nightly nutritional inputs and brain waste clearance was understood. The brain is not passively degrading. It is actively attempting to repair itself every single night, and the rate at which that repair succeeds is directly influenced by the inputs available in circulation during the repair window. You are not a passive observer of your own cognitive trajectory after 60. You are the person who decides what is on the dinner plate, and that decision has direct, measurable consequences for what happens in your brain between midnight and 3 in the morning. Now let me give you the economic calculation that makes this dinner strategy concrete. The lifetime cost of managing Alzheimer's disease from diagnosis to end of care averages $350,000 in the United States in direct medical and long-term care expenses alone, not including the unpaid caregiver burden, representing an estimated additional $200,000 in lost income and opportunity cost. Mild cognitive impairment requiring supervised living arrangements costs between $45,000 and $90,000 annually. The dinner I have described costs approximately $12 to $15 per serving, representing less than $4,000 per year in targeted neuroprotective nutrition. The gap between those numbers is not a gap in effectiveness. It is a gap in awareness that exists specifically because there is no patent and no profit margin on a dinner of salmon and spinach. If you made it this far, I want to say something directly. The fact that you watched this entire video tells me you are not someone who has given up on your brain. You suspect, correctly, that the standard answer about cognitive aging is incomplete. And now I need to ask you for something from the heart. I am one physician sharing research that a $350 billion supplement and pharmaceutical industry has no financial interest in translating into dinner recommendations. So before you close this video, do 3 things. First, if you have not pressed like yet, do it right now. Without your like, this video reaches 100 people. With your like, it reaches 100,000, and somewhere in that number is an adult over 60 who woke up this morning frightened by a memory lapse and has been told nothing can be done. Your like is the reason they find this tonight before another incomplete repair cycle passes. Second, subscribe and activate the bell. I have 35 more videos covering the neuroscience of aging that Western clinical practice has not yet translated into practical daily guidance, publishing every single week. Third, share this with one person who has mentioned memory concerns or slowed down in ways they cannot explain. Leave a comment telling me your country and the one food from this dinner you are adding starting tonight. Your action is not a number on a screen. It is the reason this channel exists. I will see you in the next video, where I reveal the specific traditional Japanese fermented food shown in 6 peer-reviewed studies to increase slow-wave sleep duration by an average of 23% in adults over 60, directly amplifying the glymphatic clearance window we discussed today. Available at any Asian grocery store for less than $6. Never once mentioned in a Western neurology prevention guideline. Dr. Jae Yoon, the science Asia has always known. Take care of your brain tonight, I mean that.